In this chapter, you will learn how gene expression is regulated through the powerful mechanism of RNA interference, or RNAi. We will examine the types of small RNAs involved, the molecular machinery that mediates RNAi, and the biological significance of these regulatory systems. By the end, you should be able to analyze and interpret how RNA molecules control gene expression at the post-transcriptional level. The discovery of small silencing RNAs transformed our understanding of gene regulation. RNA interference revealed that small RNA molecules can silence genes in a sequence-specific manner. These findings have led to new technologies in functional genomics and introduced us to eukaryotic regulatory pathways that were previously unknown. At the heart of RNAi is the introduction of double-stranded RNA, which sets off a silencing cascade. The roundworm Caenorhabditis elegans has been a central model organism in RNAi research. About 50 to 60% of its genes have human homologs, making it highly relevant to biomedical studies. Its transparent body, rapid life cycle, and well-characterized development make it ideal for genetic manipulation. The study of RNA in C. elegans has paved the way for understanding RNA-based gene regulation in higher organisms. In C. elegans, introducing double-stranded RNA can repress protein synthesis in a sequence-specific way. This can be achieved through three main methods: microinjection, feeding worms E. coli expressing siRNA, or soaking worms in a siRNA-containing solution. The success of RNAi depends on how efficiently the double-stranded RNA is delivered into the worm cells, where it can begin gene silencing. RNA interference is a post-transcriptional mechanism. Double-stranded RNA is recognized and processed, leading to the destabilization of complementary mRNA. Central to this process are Argonaute proteins and the RNA-induced silencing complex, or RISC, which includes a 22-nucleotide guide RNA. The enzyme dicer, a specialized RNase 3, plays a pivotal role by converting long siRNA into short interfering RNase siRNAs. The enzyme dicer cleaves double-stranded RNA into siRNA fragments. These are then loaded onto the RISC complex, which includes the Argonaute protein RDE1. One strand of the siRNA is incorporated into RISC, guiding it to the complementary mRNA. RDE1 then cleaves the target mRNA, resulting in gene silencing. Dicer acts independently of RISC, but is absolutely essential for initiating RNAi. This figure shows the domain structures of dicer across several species. In humans and C. elegans, dicer has a similar multi-domain architecture, including RNA's 3 domains, a pus domain, and a helicase domain. At the bottom, we also see the simpler dicer protein from Giardia intestinalis. Despite species differences, all versions of dicer carry out the same essential role, chopping siRNA into functional siRNAs. Once dicer has cleaved the double-stranded RNA, the two strands are separated. The passenger strand is discarded, while the guide strand becomes part of the RISC complex. In organisms like Drosophila, this process involves dicer 2, R2D2, a double-stranded RNA-binding protein, and formation of the RISC loading complex. This complex ensures that the correct strand is loaded and that the silencing machinery is directed to the right mRNA target. RNAi serves as an innate immune defense mechanism, especially in organisms that lack interferon systems. It can block viral replication and suppress the activation of transposable elements, which are mobile DNA sequences that can disrupt the genome. RNAi is thought to have evolved early in eukaryotic history, acting as a genome defense system against invasive nucleic acids like viruses and transposons. A remarkable feature of RNAi in some organisms is its ability to spread beyond the initial site of siRNA introduction. This is known as transitive RNAi. In C. elegans, RNAi can propagate along a gene and even move from cell to cell, leading to amplified and systemic gene silencing. This spreading effect is crucial for maintaining robust silencing throughout the organism and reveals the powerful and adaptable nature of RNAi. Transitive RNAi involves more than just spreading the silencing signal. In some organisms, the initial siRNAs produced by dicers serve as templates for generating secondary siRNAs. These secondary siRNAs are often associated with different Argonaute proteins, allowing the RNAi response to be amplified and sustained. Even more fascinating, this mechanism enables RNAi to move from cell to cell, and in some cases, it can be inherited, making RNAi a heritable gene silencing tool. The systemic spread of RNAi in C. elegans is dependent on a protein called SID1, which functions as an integral membrane transporter. Mutant worms lacking SID1 can still silence genes locally, but fail to spread the RNA signal throughout the body. Interestingly, SID1 is not expressed in neurons, which explains why neurons are generally resistant to systemic RNAi. SID1's presence enables efficient distribution of silencing signals, reinforcing RNA's systemic potential. While neurons in C. elegans are typically resistant to RNAi, researchers identified mutants with enhanced sensitivity, such as those lacking ERI1. ERI1 is a 3 to 5 exonuclease expressed highly in nerve cells. Its role is to negatively regulate RNAi, likely by degrading small RNAs and thereby limiting the duration or reach of the RNAi effect. This selective resistance suggests that RNAi can be cell type specific and that regulatory mechanisms like ERI1 fine-tune its activity. RNAi has revolutionized functional genomics by enabling targeted gene silencing. Traditionally, researchers would observe mutant phenotypes to infer gene function. But with RNAi and full genome sequences available, we now practice reverse genetics, starting with a known gene and using RNAi to knock it down, then observing the resulting phenotype. This allows for systematic study of gene function on a genome-wide scale and has accelerated biological discovery. This slide recaps the central concept of RNA interference. Double-stranded RNA leads to the specific degradation of mRNA, thereby reducing or eliminating protein production. The pathway involves dicer, Argonaute proteins, and the RISC complex. RNAi is precise, efficient, and adaptable, making it both a natural cellular process and a powerful laboratory tool for gene silencing and therapeutic applications.
Scientists have been making rapid progress in understanding RNA interference, or RNAi. Many organisms use RNAi to control genes, and it can also be used as a tool in the laboratory, and in the future perhaps as a therapy. This animation will introduce you to the principles of RNAi involving two important types of RNA molecule, small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of a cell, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by RNA polymerase II. The primary RNA transcript is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Here, ribosomes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs or double-stranded RNA is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to dicer, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaute protein. One strand of the RNA is selected and remains bound to Argonaute. This is called the guide strand. The combination of the RNA and Argonaute, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaute catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC to messenger RNAs. Usually only part of a microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs. Targeting by a microRNA can lead to messenger RNAs being degraded or translation being inhibited. Argonautes and their small regulatory RNA cofactors are found in plants, animals, fungi, and some bacteria, and their importance in a multitude of biological processes and as tools continues to be revealed.
In addition to siRNAs, MicroRNAs miRNAs are another class of small regulatory RNAs. The miRNA pathway blocks mRNA translation or promotes mRNA degradation. Each 22 nucleotide miRNA can regulate hundreds of genes, and miRNAs represent up to 4% of all expressed human genes. It is estimated that 1/3 of all human genes are under miRNA regulation. Most miRNAs originate from primary miRNA transcripts, piRNA synthesized by RNA polymerase II. PiRNAs possess 5 methylguanosine caps and 3 poly-A tails, just like typical mRNAs. These transcripts are processed by the microprocessor complex, which contains an RNase 3 enzyme and a double-stranded RNA binding protein. After nuclear processing, the precursor miRNAs, pre-miRNAs, are exported to the cytoplasm, where they are further cleaved by dicer 1 into a 22-nucleotide miRNA duplex. One strand is then incorporated into the miRNA complex, allowing it to guide gene regulation. If a miRNA is encoded within an intron, the lariat is cleaved before export from the nucleus. For non-intronic miRNAs, the microprocessor complex acts first. Once in the cytoplasm, dicer 1 cleaves the pre-miRNA and loads it onto an Argonaute protein to form miRNAs. If the miRNA has complete complementarity with its target, the mRNA is cleaved. If there is incomplete pairing, miRNA binds to the 3' untranslated region and inhibits translation without degradation. This slide offers a summary of the miRNA regulatory pathway, emphasizing its post-transcriptional control over gene expression. miRNAs can suppress protein output either by degrading mRNAs or by stalling translation. The versatility of miRNAs and their widespread presence across human tissues highlight their importance in development, differentiation, and disease, including many types of cancer and neurological disorders.
Two families of small regulatory RNAs called microRNAs and small interfering RNAs or siRNAs regulate the stability and translation of mRNAs in eukaryotes. Both classes of small RNA associate with a family of proteins known as Argonaute, but differ in their origin, processing pathway, targets, and mechanism of action. We will first focus on microRNAs and then point out the similarities with and differences from siRNAs. MicroRNAs derive from actual genes found in genomes across the lineages of multicellular animals and plants. These RNAs begin as primary transcripts or pri-microRNAs and are usually transcribed by RNA polymerase II. The pri-microRNA transcript folds into a stem loop structure that generally has some unpaired nucleotides, and single-stranded extensions are found at both the 5' and 3' ends. This structure serves as the initial substrate for processing by DROSHA, a nuclear RNase 3 enzyme. DROSHA interacts with a specialized RNA binding protein called DGCR8 in humans to form what is termed the microprocessor complex. This complex performs a cleavage reaction that removes the 5' and 3' extension and liberates a 60 to 70 nucleotide transcript known as a pre-microRNA. The pre-microRNA generated by the microprocessor complex is recognized by a nuclear export factor called exportin 5, which transports the pre-microRNA to the cytoplasm for subsequent processing. In the cytoplasm, a second endonucleotic cleavage reaction, generally referred to as dicing, is catalyzed by dicer, another RNase 3 enzyme. Dicer is found in complex with a double-stranded RNA binding protein called the TAR RNA binding protein 2, or TRBP2. The product of this second cleavage reaction is called the mir-star duplex. It carries 5' monophosphates and 3' overhangs of two nucleotides, which is not fully complementary. The two strands of this duplex RNA are called the guide and passenger strands. Once the mir-star duplex has been generated, it is loaded into the Argonaute protein that is dedicated to microRNA-mediated silencing reactions. If the duplex is loaded in an appropriate orientation, one of the RNA strands, called the mir guide strand, is retained, while the other strand, the mir-star passenger strand, is selectively removed. The guide strand will ultimately be involved in determining which target RNAs will be silenced. This process of retaining one strand and removing the other is called sorting. For imperfect mir-star duplexes, it is not certain how the passenger strand is identified and selectively removed, though the action of a helicase may well be helpful. Argonaute proteins charged with their guide microRNAs are referred to as the MI-RISC complex. The complex is now ready to bind its target RNA and promote gene silencing. Argonautes bound to microRNAs typically identify sequences with imperfect complementarity in the 3' UTRs of mRNAs. The most important pairing region for the microRNA is referred to as the seed sequence, which generally encompasses nucleotide positions 2 to 8 of the microRNA species. The absence of extended duplex formation allows the 3' end of the microRNA to remain securely bound to the PAS domain of the Argonaute protein. As a consequence, the Piwi domain of the Argonaute protein is generally not positioned appropriately to cleave mRNAs that are targeted by microRNAs. Once bound to targets, MI-RISCs appear to recruit additional factors, including in particular a protein that contains multiple glycine tryptophan repeats, which is known as TRNC6 in humans. This protein is thought to be involved in repressing translation and in destabilizing the mRNA through mechanisms that remain poorly understood. What is clear is that the repression mechanism does not depend on the sliceal activity of the Argonaute protein. In contrast to microRNAs, siRNAs are not generally encoded in the genome as specific genes. Instead, siRNAs are derived from double-stranded RNA that comes from several different sources. Endogenous duplex RNA can arise from the normal transcription of genomic loci that have extensive hairpin structures, or from the annealing of sense and antisense RNAs that have both been transcribed from a given locus. Exogenous sources of double-stranded RNA include viral RNAs and duplex structures that have been synthetically introduced into cells by scientists for experimental purposes. Irrespective of their origins, duplex RNAs become substrates for an RNA processing reaction akin to the one described for the microRNAs, but without the first nuclear drosha-dependent cleavage. Only dicing by the dicer enzyme is needed. The cytoplasmic enzyme sequentially cleaves the long duplex structures approximately every 20 to 25 base pairs. The cutting frame is set by interactions between the double-stranded RNA and the Pas region of the dicer protein. The products of the dicer reaction are short duplex RNAs, similar to the mir star duplexes, but fully base paired along their length. Once the SIR star duplex has been generated, it also is loaded into the appropriate Argonaute protein, and the SIR star passenger strand is selectively removed. For these fully complementary SIR star duplexes, the endogenous endonuclease activity of the Piwi domain of the Argonaute protein catalyzes cleavage of the passenger strand. The cleaved strands are then easily unwound and released. This mechanism makes use of the RNase H-fold of the Piwi domain and acidic residues that are poised in the active site for such catalysis. The resulting single-stranded siRISC complexes can now scan for their fully complementary target RNAs. In contrast to the situation with the miRISC complexes, fully complementary binding to the target RNAs leads to the 3' end of the siRNA being displaced from the Pas domain of the Argonaute as extended duplex nucleic acid forms. This results in conformational changes that activate the cleavage activity of the RNase H Piwi domain. The cleavage reaction is referred to as slicing. Products of the slicing reaction are thought to immediately become targets for the exosome and other standard RNA decay pathways in the cell.
The third class of silencing RNAs is piRNAs, or Piwi-interacting RNAs. These are slightly longer, 25 to 30 nucleotides, and possess 2 O-methyl modifications at their 3 ends. They bind to members of the Piwi subfamily of Argonaute proteins. piRNAs are primarily found in the nucleoplasm of germ cells and adjacent somatic tissues. Their main role is to suppress transposable elements, helping to maintain genome integrity in the germline. piRNAs likely perform a similar function in vertebrates as well.